So welcome everyone. Sorry for those of you on Zoom, but yes, lunch was actually quite good. So anyway, so just as a word of introduction, I'm Neville. For those who don't know me, I'm Neville Calleja. I'm from Malta, as Mariella was saying. I'm full professor in public health at the University of Malta, and I'm also head of department at the what we call the Directorate for Health Information and Research within the Ministry of Health in Malta. Um, and we're responsible for official statistics, basically, for the whole country. But also, within this directorate, we house the secretariat for what we call the Health Ethics Committee, which is the nationally legislated competent authority for the ethical assessment of clinical trials and has been so for almost 20 years now. Also, part of my history, I was also an assessor for clinical trials applications at the Medicines Authority, so that's on the scientific side, so to say, of the assessment of clinical trials with the Malta Medicines Authority. Um, uh, this is when we started up, basically when we joined the EU. So again, I'm talking around 19 years back, so things have evolved, of course, since then, nonetheless. So the session today, I've been asked to talk to you about interpreting and reading clinical trials. I'm also aware that many of you are already exposed to some extent to clinical trials or sort of to reading research, so to say. So I try to focus less on the traditional critical appraisal skills, even though I'll just make a very brief reference to that. But I wanted to go beyond those, beyond what our typical critical appraisal toolkits expect us to do when evaluating a clinical trial. So, again, just in case, I know cost actions are by definition bringing together professionals from a multidisciplinary area, so maybe some of you are indeed not so exposed to clinical trials. So a very quick couple of slides. When we say clinical trials, we're typically looking at a highly controlled experiment, okay, where you are recruited into a study, assigned into a particular treatment arm, be that the intervention group, the control group. That's the simplest form, of course, because you can have multiple groups or multiple arms, as we call them. Okay? And then obviously we're going to observe the outcome of our intervention or lack thereof on the study subjects and compare ultimately what was the effect of the intervention. I think that is the most simplistic explanation of a clinical trial. I've put some random, well, not random, words down there in a word cloud. I'm hoping that many of you are already familiar with these terms: double blind, single blind, randomized, as in every person who's recruited into the study is randomly assigned to any of the groups. Okay? Crossover, parallel, controlled, or uncontrolled. So I'm not going to delve more into these terms because I know many of you are already fairly familiar. Just also to remind you about what we call the hierarchy of scientific evidence, which, at least based on my personal experience during the last 2 years of COVID pandemic, it seems many even healthcare professionals have forgotten this. Okay? So just to remind you that a case report, okay, just before coming here, because for my sins, I follow some of the top misinformers in the world on Twitter, just to know what's happening. And the latest is that Madonna was admitted to hospital. I'm not sure if you've received this. And obviously, people are attributing this to her vaccine, and nobody actually asked whether she does actually have pneumonia, as in fact she does. Anyway, so case reports are at the bottom of that list. And then as we go further up, we're seeing more rigor coming into these studies, right? And clinical trials are almost at the top, almost, not at the top, okay? Because then at the top we have meta-analyses and systematic reviews that typically bring together a number of trials, a number of studies, okay? So we're not just basing ourselves on one study, but we're bringing many together. In some of the depictions of this, you also see another layer, which are the sort of reviewed opinions, bringing together clinical trials and evidence. So anyway, whichever you like. So, but roughly there we are, okay? So randomized clinical trials are considered to be at the top of the hierarchy of evidence. And none of this, of course, actually enters the hierarchy of evidence. Okay? So, moving on to the concept of critical appraisal. Again, I'm not going to dwell too much on this, but I understand this is a training school, so I felt we should just touch briefly on this. Okay, so when we say critical appraisal, it's not a tool. Some of my students think that critical appraisal is there for them to find a fault with a study and throw it away. No. Critical appraisal, as the word implies, tries to help us form an objective overview of that paper. Not just how valid it is or how trustworthy the results are, but also how applicable that evidence is to our particular context. Okay? So if I'm reading a study about, I don't know, the effect of, I know I'm deviating away from gynecology here, but the effect of ACE inhibitors on the black skin population, which we know there are some peculiarities, and I don't seem to have that many black-skinned patients in my practice, then maybe, its value for my practice is a bit limited. Okay? This is, at least this is the toolkit I usually use with my students. This is just one of them. This is for clinical trials. I put a QR code there for those of you who are interested in checking these toolkits out. They were developed by the NHS in the UK. They're fantastic. They're all following the same structure. You roughly get 10, 12 questions for 6 types of studies, and they all follow the same structure. They're simply adapting for the different types of studies. Cohort study, case control study, qualitative studies, okay? But the points, the concepts are always the same. And basically the way these toolkits work is if you see that a study is failing miserably in the first questions, you can actually stop and maybe discard the paper because clearly it's not valid and therefore you don't proceed. But I must admit again, I discourage my students from doing that, especially at the initial stages, because one risks that they're throwing away papers that might have some value, okay, but they're just being dismissive. So this is something, again, that maybe you can come back to at a later stage. But I think the 2 most important areas of the critical appraisal toolkits is what we call the PICO tool, or whatever, PICO principle. PICO stands for participants or population, okay? So basically, in the paper I'm appraising, this needs to be fairly obvious. Who is being included in the study? As simple as that. Which is the population? Who are the patients? I is for intervention or indication, okay? So what are we doing this experiment for? What test? What's the management? What's the exposure we're researching? C is for comparator or control, okay? So we need to ensure that the clinical trial does include some kind of comparison or control. And it goes beyond that, okay? Are the 2 groups being equally exposed, right, to the same aspects? So everything else needs to be the same apart from the intervention or exposure of interest. What are the patient, then the outcome, of course, is the O. What are the consequences, the outcomes we are studying? So again, I usually tell my students, guys, these 4 points should be pretty self-evident, at least, if not by the end of the abstract, by the end of the introduction of the paper. If they're not, we're on marshy ground, so to say. Okay? The other principle that we usually look for is what we call the consort diagram, and Camilla has nothing to do with this. Okay? The queen consort has nothing to do with this. So this is known as the consort flow diagram. I'm sure, again, most of you are familiar with this, but basically this gives us a graphical depiction of where the patients have ended up. Okay? How many have we approached? How many were eligible? How many were ultimately recruited, randomized, followed up, and analyzed? This, again, should be transparent information in the clinical trial you're assessing. I think these, personally, I feel these are probably two of the main thrusts of the critical appraisal toolkit we discussed earlier. If these two are robust, then I think it's worth racking our brains and trying to understand the results of the paper, which can sometimes be challenging, that I admit. And once we do that, once we find that the results are, clearly significant and something to take note of, then we might as well look at how applicable they are to our situation, to our funding, to our patient population, whatever it is. Now, the other points that are maybe not that explicitly included in the critical appraisal tools that we use. One of them, which is quite important, is missing data. Okay? We need to have, again, the consort diagram earlier helps us to appreciate this quite well. Okay? Do we have missing data in the trial? No? No problem. Yes? Which one of these three applies? Is it missing completely at random? Which happens. Okay? Not everybody who starts a trial will actually finish it. That's normal, that's life. Okay? If that is happening pretty much equally on both sides of the trial, then that's fine. That's kosher, it's cool. All right? No problem, no bias. If it's missing at random but not completely, what we mean by that is that we might have some of the missing data being dependent on the observed data. Let's give an example, all right? I remember back in our medical student days, let's say, well, we've been 25 years now out of medical school, things were a bit less ethical at the time, but I remember some of our colleagues were involved in a bioequivalence study back home for, oh God, my TB treatment knowledge is getting a bit fuzzy now, but was it rifampicin? The one that makes your sweat turns red, right? Rifampicin, I think it was. And anyway, some of us were assigned to the new rifampicin, anyway, and some to the original, to the generic and to the original. And for some strange reason, those assigned to the generic rifampicin, the new one, had more staining. And this was June. June in Malta is warmer than Sofia. Okay? So it was very easy to spot who was on the generic rifampicin and who was on the original. Needless to say, those of us who were on the generic had a higher likelihood of quitting the trial. Okay? That said, that is missing at random, not missing completely at random, because it was something that could be captured. Okay? On the other hand, if I'm doing a study that maybe there's a risk factor which is relevant to my outcome, which I'm not capturing information about, and I'm actually having some problems in recruiting from that subpopulation, which I can't identify, then that becomes a problem. And that becomes then the last category, just missing. So in the second category, we can typically adjust, for example, using weighting or multivariate analysis. Okay, I'm not going to go into detail on this. But when it's missing data for which we have no observation, then frankly, excuse my French, we're screwed. And this study might actually be systematically biased. Okay? When we see an effect, is it always significant equals yay? Not necessarily. Okay? First of all, if we're dealing with small samples, we could actually have a situation where we get an accidental event. All right? So remember, statistics is not a precise science. It's actually an approximative science. Okay? So small samples may be dangerous because we might think we've detected something, but what we've really detected is something accidental. Okay? So that's one issue. The other issue would be bias due to factors other than the intervention. And in fact, one of the first things we usually do when analyzing clinical trials is that we make sure to compare the groups, the arms, in terms of any other factors that may confound the effect of our intervention. Okay? So basically, once we exclude these two, then we can conclude we might have a real treatment effect. Speaking of biases, again, quick run through. Bias can arise from a variety of sources. I don't need to go into that. But any source of bias can be either random or systematic. Random bias, for example, I don't know, let's say I have somebody out there measuring blood pressure. There will always be an element of error, okay? Again, I like to tell my students if I send out two nurses out there for a blood pressure study, one of them is a bit more deaf than the other one. I send the deaf one to the north, I send the better hearing one to the south, I'm going to find a difference between north and south which does not exist, okay? That would be systematic bias. How can I address that? Knowing that one is deafer than the other, I could give half the north to one, half the south to the same one, half the north to the other one, half the south to the other one. And that makes it random because it will even out. Okay? Again, this is, for example, one reason why we tend to go for electronic means of blood pressure measurement, even though we always say that it is less accurate than, you know, the mercury, sphygmo, and stethoscope, etc. But that can be calibrated. So basically, if we're facing any bias, we just need to make sure that we keep it random. Okay? And that's important when, again, analyzing and interpreting a clinical trial. Okay? So if it's unavoidable, we just need to make sure that everybody is exposed to that bias equally. Another point on conflict of interest, this is not always declared. Okay? So it's most of the time it's expected of someone to declare conflict of interest, but it's still voluntary. Okay? Again, if you're caught out, it's worse, of course, but very few get caught out. So financial conflict of interest, this study has been sponsored by Philip Morris International. Okay? Typically for, if you find a smoking study that says there are benefits, look at the end and you typically find sponsored by Philip Morris International. I think you can add cannabis studies now to that as well. That is typically declared. So this is direct funding of the study, grants, etc. Okay? But we have other conflicts of interest, which I think you can understand, which may not be so clear and are typically not declared. It could be investigator-based. Okay, so I'm not getting money for this study, but the company is paying the education of my daughter. Okay? So there are some, they're not, huh? Not in my case. But that would be investigator-based. Institution-based. My university has a wing being built by pharma company XYZ, and lo and behold, I'm studying a product which is manufactured by XYZ. Okay? I'm not getting anything myself, but my institution might be. And lastly, IRB-based, institutional research boards based. Now, this might sound shocking, but in my 25 years of experience working in clinical trials, I have come across, I will not identify neither the company nor the country, but I have come across a clinical research organization which admitted to me that they actually set up the ethics committees in a particular EU country. Okay? So there are such conflicts of interest sometimes at institutional research boards level. These might not be that visible. Can I actually identify these kind of biases? Very difficult. But it's always good to have a healthy level of paranoia, basically. Okay, if you see something that's strange. So, are clinical trials the panacea that sometimes we think they are? Okay, they definitely have advantages. Sorry, the slide I realize is a bit tiny. So it's definitely an efficient method to observe the impact of an intervention. Randomization takes care of a lot of other variables. Okay, so we do decrease a lot heavily, so to say, the incidence of bias and confounding factors. And we definitely get a lot of priceless information on the efficacy of a drug along with benefits and risks. Now here I must add one pet irritation of mine, that when we get a drug approved by EMA or our national regulatory authorities, nobody gives a hoot about cost-benefit. Okay? So I've actually spent almost around a year as Chief Medical Officer back home, and part of my work was being faced with applications. Dr. So-and-so is applying to get this oncology treatment, which costs $750,000 a year for a patient to gain 3 months of life. It's available on the market. The oncologist would not take the responsibility of refusing it. But as Chief Medical Officer, if I approve that drug for that patient, it means I am depriving, 7 kids from getting treatment of $100,000 each. Because ultimately money does not grow on trees, definitely not within my government. Okay? So that's another issue that clinical trials, unfortunately, most of the time don't go into, the health economic aspect. Okay? So, as we said, yes, this information is obviously critical for setting public health priorities and clinical decision making. Okay, so what does not happen at EMA typically happens then at funding agencies or health insurances within the countries. So EMA makes them look bad then. The disadvantages, we get a lot of this criticism. A clinical trial might not be a good reflection of the real world out there. Okay? Patients sometimes are not understanding, they don't follow instructions, whatever. Okay? Clinical trials are indeed, given the restrictions, given the controlled environment, they're very expensive to perform, time-consuming, and obviously much more complex than observational studies. What's the effect of this? That the final price tag on that box of medicines will obviously go up. Okay? Or at least we can give an excuse to the company to push up the price tag on that box of medicines. Then this is important. There's the ethical aspect. When we approve a clinical trial application as an ethics committee, we're always weighing these two sides. We are basically depriving half the study population from the new drug, so we need to have enough doubt to do that. On the other hand, we are exposing half of the population to the new drug, so we need to have enough faith in the product to do this, all right? Once this disrupts then we have a problem. Okay? And we have to maybe stop the trial. The recruitment of patients in certain situations, particularly relevant to you guys here, given you're dealing with rare gynecological cancers, recruitment of patients for rare diseases can be particularly challenging. And in fact, this has been on the agenda of also international organizations like Eurordis, etc., to facilitate this as much as possible. EMA also has, and most other drug regulatory agencies also have different requirements when it comes to orphan drugs, that is, drugs for rare diseases. And usually their requirements for the clinical trials are less rigorous. And therefore, if we're looking at clinical trials for rare diseases, we might have to live with making certain compromises on the rigor of the study. Okay? And this is important. Statistical significance may not always translate into clinical significance. This is particularly relevant for markets such as hypertension, where we get products being launched on the market, every minute, and everybody is claiming they have an additional significant 2 millimeters of mercury advantage on their competitor. Yeah, right, okay, so I'm going to let go of the product that I know even better than my wife. Okay? To switch to a new product which gives me 2 millimeters of mercury advantage, as if. Okay, and lastly, the issue of publication and sponsorship bias. Again, I don't know if any of you sit on regulatory authorities and you receive dossiers from companies to approve a drug. Remember, what you see in the dossier is a sub-selection of all the research that they have done for that drug. Okay? So I think somebody actually estimated that we don't see more than 20% of generated evidence in a dossier, in an officially submitted dossier. Okay, so there's always a bit of choosing and selection in what goes into that dossier. So again, once again, a healthy dose of paranoia helps. So if you spot something, you might wish to question that, all right? These typically, unfortunately, then come out after marketing authorization. And I'm sure you can all remember a product or two that came out with a lot of hype, and then after 1-2 years on the market, some independent researcher put a lot of studies together, did a meta-analysis, lo and behold, there's a serious side effect, cardiac events, etc., and it's withdrawn from the market. Okay? So that's important. Most negative outcome research does not actually make it to publication. What helps, admittedly, is the culture of registering clinical trials. And I'm not sure if you're familiar with this. This started in the States with the clinicaltrials.gov website, and many actually register on that, even from Europe. There's now also an equivalent version in the EU. If I'm not mistaken, it's clinicaltrialsregister.eu, where basically whoever starts a clinical trial, even our students sometimes, typically register their research there. So again, if you look through these websites, you can find what clinical trials are happening and then ask, hey, was this published? So like that, one can follow up on what actual research has started, as long as it's registered. So this mitigates a bit the whole issue. And I think that brings me to the end of my session. I'm not sure if you want to take questions now or later.